Hello, my name is Iman Khalid, and this is group number 13. We are going to talk about proteomics and bioinformatics under the supervision of Dr. Abdelbary El Prince. Introduction. Proteomics. It is a large-scale study of proteins, their structures, functions, and interactions within biological systems. It is a branch of molecular biology that focuses on the identification, characterization, and quantification of all the proteins present in a given sample, such as a cell, tissue, or organism. Proteomics involves the use of various techniques such as mass spectrometry, protein microarrays, and bioinformatics to analyze and interpret the vast amount of data generated from protein analysis. This information can provide insights into the underlying mechanisms of biological processes, including disease progression and drug discovery. Proteomics can be applied to a wide range of fields, including medicine, agriculture, and environmental science. For example, it can be used to identify potential biomarkers for disease diagnosis, monitors the effects of drugs on protein expression, and analyze the protein content of food crops. Types of proteomics. Number 1. Expression proteomics is a novel approach that studies the quantitative and qualitative expression of proteins. It aims to specify the difference in protein expression between two conditions, such as patients and controls. Expression proteomics experiments are usually used to study the patterns of protein expression in different cells. For example, a tumor tissue sample is compared to a normal tissue sample to identify differences in the levels of proteins. In addition, it can identify disease-specific proteins and new proteins in signal transduction. Variations in protein expression which are present or missing in tumor tissue compared to normal tissue are detected using 2DE and MS techniques. Number 2. Structure proteomics. It specifies all protein interactions such as membranes, cell organs, and ribosomes in the mixture. Nuclear magnetic resonance spectroscopy and X-ray crystallography are used in structure proteomics to determine the three-dimensional structure and structural complexities of functional proteins. The study of the nuclear pore complex is an example of structure proteomics. And number 3, functional proteomics. This type of proteomics studies the protein functions, molecular mechanisms in the cell, and determines the protein partners interaction. In particular, it investigates the interaction of an unknown protein with partners from a specific protein complex involved in a particular process. This may indicate the biological role of the protein. In addition, the elucidation of protein-protein interactions in vivo can lead to a comprehensive descriptions of cellular signaling pathways. What is the methods of proteomics? 3 methods for separation of complex protein or peptide samples. Number 1. Denaturating polyacromide gel electrophoresis (PAGE) or sodium didecyl sulfate polyacromide gel electrophoresis (SDS PAGE). The ionic detergent SDS denaturates and binds the proteins to make them uniformly negatively charged. And number 2. Two-dimensional gel electrophoresis separates proteins based on both isoelectric point and molecular weight. Number 3. High-performance liquid chromatography (HPLC) used to separate the components in a liquid mixture. What is the applications of proteomics? Number 1. Oncology. Application of proteomics in cancer, oncoproteomics. Number 2. Drug discovery, a complex process with many different stages, chemical, functional, and clinical proteomics-based approaches. Number 3. Biomarker discovery. A biomarker is an accessible pointer of a normal or abnormal biological state in the body. In clinical settings, cancer development and its response to therapy are measured by cancer biomarkers. Number 4, leukemia proteomics was used to discover many leukemia biomarkers that could determine types of leukemia. And at the end, indeed, bioinformatics is a very important science in our lives. And thank you for your listening, and thank you.